Now we move to microarray. In each type of cell, like a muscle cell or a skin cell, different genes are expressed, turned on, or silenced, turned off. If the cells that are turned on mutate, they could, depending on what role they play in the cell, trigger the cell to become abnormal and divide uncontrollably, causing cancer. One way they do this is to use a DNA microarray to determine the expression levels of genes. When a gene is expressed in a cell, it generates messenger RNA, mRNA. Overexpressed genes generate more mRNA than underexpressed genes. This can be detected on the microarray. Red spot shows that gene was strongly expressed in cancer cells. Green spot shows that gene was strongly repressed in cancer cells. Yellow spot shows gene was neither strongly expressed or strongly repressed in cancer cells. Black spot indicates that none of the cDNA has bonded to the DNA in the gene. The first step is to collect healthy and cancerous tissue samples. Once the tissue samples are obtained, mRNA is isolated from the samples. mRNA is color-coded with fluorescent tags and used to make DNA copy. The mRNA from the healthy cells is green, meanwhile in abnormal cells is red. How DNA microarrays work? DNA copy that is made, called cDNA, is then applied to the microarray. The cDNA binds to complementary base pairs in each of the spots on the array, hybridization. Each spot will appear red, green, or yellow when scanned with the laser. There are 3 applications in microarray, which are 1. Cancer, 2. Antibiotic treatment, 3. Oral pre-cancer. Cancer. Genomic DNA is fluorescently labeled. Array-based comparative genomic hybridization used to map genetic abnormalities in a wide range of tumors. Gene expression data can identify groups of cases with significantly different outcomes. Antibiotic treatment. Increase in number of resistant bacteria led to failure antibiotics. In oral cavity anaerobic bacteria, they are not easily culturable. DNA microarray analysis helped to outlast the viability of the bacteria and a diagnosis can be made. Oral precancer. Leukoplakia or white lesions of the oral cavity may result from a myriad of reversible conditions. Identification of gene expression profiles allow to differentiate harmless white lesions from precancerous lesions. Early diagnosis and management of oral cancer is correlated with increased survival. Single strand of DNA of gene of interest can be immobilized in a grid or array, which then will be either glass slide, quartz wafer, and a nylon membrane. For example, in tumor biopsy, mRNA is extracted, labeled and hybridized with the array.
Created using Powtoon.
What is expressed sequence tags?
EST are fragments of mRNA sequences derived through single sequencing reactions performed on randomly selected clones from cDNA libraries. ESTs represent partial sequences of cDNA clones 300 BP to 700 BP. EST pre-processing reduces the overall noise in EST data to improve the efficacy of subsequent analyses. Vector contamination is prevalent in ESTs and need to be clipped before EST are clustered. Purpose of EST clustering is to collect overlapping ESTs from the same transcript of a single gene into a unique cluster to reduce redundancy. 2 approaches for EST clustering: stringent and loose. Stringent conservative clustering method uses single-pass grouping of ESTs resulting in relatively accurate clusters. It generates shorter sequence consensuses. Loose is liberal and repeats low-quality EST sequence alignments many times to generate less accurate. Loose generate longer sequence consensuses. Once consensus sequences, putative genes, are obtained from assembled ESTs, possible functions can be assigned through downstream annotation. Achieved via database similarity searches. employing familiar freely available tools and databases. First step in translating EST sequences is in identifying the protein coding regions or open reading frame from consensus EST sequences, or predictor designed specifically to identify protein coding regions in EST-derived sequences. Once a putative polypeptide is obtained, its function can be predicted by matching against non-redundant protein sequence, motif and family databases using an integrated tool such as InterProScan. Protein sequences are better templates for functional annotation. Gene discovery, aid gene structure identification, establish the viability of alternative transcripts, guide SNP characterization and facilitate proteome analysis. Providing markers for mapping, providing candidate disease genes, and analyzing gene expression. A complex multiple-step process that makes use of several distinct programs. Clustering the sequence based on similarity and then assembling them to produce contigs. Purpose: reduce the search space when performing similarity searches. Estimating the number of genes represented in the dataset. Use when there is a large-scale sequencing project in which thousands of EST are generated daily. Can store, organize and annotate using an automated analysis pipeline. Automatically clean, cluster, assemble, generate consensus sequences, conceptually translate and assign putative function.
Created using Powtoon.